Hello students, in this video we are going to see the last part of class 6 basic science first chapter Cascades of Life. The last part of this chapter deals with plant cell and comparison between animal and plant cell. We have learned about animal cell. Now we have to think about plant cell. So let us examine a part of a plant. So at first we have to do an experiment, that is observe the onion peel through a microscope. So for that what we should do, we should prepare a slide. So here is the box. Method of preparing slide. Remove the dry outer layers of onion. Then peel off the thin layer from the fleshy part. Keep this in water in a watch glass. After staining in safranin solution, place a small portion on a slide by using a brush. Cover it with cover glass. So this is the method of preparing slide. Okay. Now let us see how to prepare this slide in the video.
A stained temporary mount of an onion peel. To do the experiment we require cover slip, glycerine, glass slides, onion, watch glasses, distilled water, safranin solution, forceps, brush, needle, compound microscope, blotting paper, and a dropper. Procedure. Pour some distilled water from a bottle into a watch glass. Take half a piece of onion and remove leaves from the onion piece. Remove a piece of transparent onion peel with the forceps. Put the epidermis into a watch glass containing distilled water. Take some safranin solution using a dropper. Put a few drops of safranin solution into another watch glass. Using a brush, transfer the peel into the watch glass containing safranin solution. Keep the epidermis for 30 seconds in safranin solution so the peel can become stained. Take the peel from the safranin solution using the brush and put it in the watch glass containing distilled water. Using a dropper, take some glycerine and put 2 to 3 drops of glycerine in the middle of a dry glass slide. Transfer the peel to the slide containing glycerine using the brush. Take a cover slip and place it gently on the peel with the help of a needle. Remove the extra glycerine using a blotting paper. Take the glass slide and place it under the compound microscope. View the slide under the compound microscope. Observation. A large number of regularly shaped cells are lying side by side. Each cell has a distinct cell wall. A distinct nucleus is present on the periphery of each cell. Lightly stained cytoplasm is observed in each cell. A large vacuole is present in the center of each cell, surrounded by the cytoplasm.
Now we have seen the cells of onion peel. Haven't you observed guard cells in your lower classes? We have learned about stomata and guard cells in our 5th standard. Now let's see the picture of guard cells. Don't you see other cells also with the guard cells in the given picture? Yes, along with guard cells, you can see some other cells also. Are the guard cells, the other cells in the leaves and the cells in the onion peel same in shape and size? Now we have to find out whether these cells are same in shape and size. Okay, so let us see a video.
This is the upper surface of the leaf, which contain upper epidermis, means upper epidermal layer, and this is the lower surface of the leaf, which contain lower epidermal layer. And what is epidermal layer? Epidermal layer, which is the outermost transparent layer present on the surface of the leaf. First of all, what we have to do? We have to separate, means peel out the small part of the epidermal layer present on the surface of the leaf, and this part is called epidermal peel, in which stomata are present. For this, what we have to do is to tear the leaves from the leaf margin and then fold the teared portion of the leaf and then pull the folded portion of the leaf in the opposite direction of the venation carefully in such a way to separate the epidermal layer. You can see in the video the thin transparent layer, and this thin transparent layer is called epidermal layer, in which stomata are present. Completely tear and separate that part of the leaf containing the isolated epidermal peel, and here we have taken the epidermal layer of the lower surface of the leaf, because in the lower surface of the leaf large number of the stomata are present as compared to the upper layer. And now we are again showing this step of separation of epidermal layer, means epidermal peel, slowly. First, what we have to do? We have to tear the leaf from the margin and then fold the one part of the leaf and then again pull the folded portion in the opposite direction to the venation of leaf slowly, and now you can clearly see the thin transparent layer, and this thin transparent layer is the epidermal layer. And we have to use these thin transparent epidermal layer to observe the stomata present in it under the microscope. Completely remove the excess part of the leaf, and the part of the leaf which contain the epidermal peel, we have to cut and separate this epidermal peel from the leaf, and for this place the leaf part containing the epidermal peel on the slide and cut and separate this thin transparent layer from the leaf with the help of razor blade, and then transfer this small portion of the epidermal layer into the watch glass containing plain water and with the help of brush, and then transfer this section to the dilute saffron solution for 30 seconds to stain the peel, means to color the stomata, and after 30 seconds remove this section, means epidermal peel, from the stain and then again transfer into the watch glass containing plain water to remove the excess stain from the peel with the help of brush. And after removing the excess stain from the epidermal layer, then transfer this epidermal layer, means epidermal peel, on the center of the clean and dry slide with the help of brush in such a way to avoid the folding of the epidermal peel. And after this, add 1 or 2 drop of glycerin solution with the help of dropper on the epidermal peel, and then cover this epidermal peel, means epidermal layer, with the help of clean cover sleeve. Hold the cover sleeve from the side and place it vertically over the slide and hold it with the help of needle, and then slowly cover the peel in such a way as shown in the video to avoid the formation of air bubble. And now the slide is ready to observe under the microscope. Here we are observing the epidermal layer in 100x magnification, means we are observing in 10x objective lens and 10x eyepiece of compound microscope. Here you can clearly see the number of small pores scattered between the epidermal cell, and these small pores are called stomata. Then we are observing the same slide in the 45x objective lens and 10x eyepiece, and here you can clearly see the stomata embedded between the epidermal cell, and through this the stomata gaseous exchange takes place in the plant. And this stomatal pore, which is called stoma, is surrounded by 2 guard cells, as you can see the diagram which we have labeled, and these guard cells control, means regulate the opening and closing of stomata. Guard cell is surrounded by the epidermal cell, as you can see in the diagram, and by the number of the epidermal cells surrounded to each stoma and also by its pattern, we can identify which type of stomata is present in that particular leaf. And this is how we prepare the temporary slide of the stomata to observe it under.
Now look at this picture. How many types of cells you can see? You can see 2 types of cells here. One is like kidney shape and other one is like the cells which you have seen in the onion peel. Now you have seen the cells of onion peel and the different cells in leaves also. All these cells are different in shape and they have their own peculiarities. Now apart from these 2 cells, let me show you the cells in stem. These cells also have their own shape and their own peculiarities. Okay, so different cells have different shape and have their own peculiarities. Now let us go in detail about the parts included in a plant cell. Okay, so before that, let us see an animation video of the plant cell. Here is the picture of a plant cell. Let us see the parts of a plant cell. First one is nucleus. Nucleus is the center part of the cell and it controls all the activities of the cell. Second part is chloroplast. What is chloroplast? It allows plants to capture the energy of the sun in energy-rich molecules. Chloroplasts are those which allow plants to capture the energy of sun during photosynthesis. Next one is cytoplasm. Cytoplasm, as we said earlier, it is a matrix filled inside the cell. Next one is cell wall. The outer wall-like structure is known as cell wall. Next is vacuole. We have already told that there will be vacuoles in both animal and plant cells. Vacuoles in plant cells will be bigger than those in animal cells. Now next part is cell membrane. Cell membrane is the covering of the cell which protect the inner parts of the cell. Okay. So these are the parts of the plant cell. Now we have studied about both animal cell and plant cell, and we have also seen the parts in animal cell and plant cell. Now let us compare the figures of animal cell and plant cell and find out what are the differences. While comparing the parts of animal cell and plant cell, at first we'll see which are the common parts in both animal and plant cell. Nucleus, cytoplasm and cell membrane are the common parts seen in animal and plant cell. Cell wall and chloroplast is present only in plant cell. Both these parts are absent in animal cell. Vacuole of plant cell is larger than animal cell. Okay. So these are the differences between plant cell and animal cell. Now let us see once again what are the functions of different parts of a cell. Cell membrane, that is the outer covering of the cell, and it is present in both animal and plant cell. Next one, cytoplasm, matrix or liquid filled inside the cell, present in both the cells. Next, nucleus, center of the cell, present in both cells. Vacuole, storage of cell waste, present in both cells. Mitochondria, energy house of cell, present in both cells. Cell wall, provides strength made of cellulose, that is only present in plant cell. Next is chloroplast, helps in photosynthesis, present only in plant cell. So these are the different parts of the cell and their functions. Okay. So in this chapter we have learned about different types of cells, and we have learned that millions of cells are there in the human body, and cells are the fundamental units of life. And we have also seen about animal cell, plant cell, and differences between both. Okay, thanks for watching.